Starting here looking at glucose catabolism in the absence of oxygen. We're looking at what happens if we don't have oxygen readily available. And so if there's not enough oxygen around, oxygen cannot drive the electron transport chain. And so it's going to back up. Remember, oxygen is the final electron acceptor to keep that chain moving. And so in the absence of oxygen, the Krebs cycle backs up and the electron transport chain backs up. Glycolysis can continue. Remember, glycolysis generates ATP via substrate level phosphorylation, so it doesn't require oxygen. And so glycolysis to some extent can continue to occur only if the NADH that's produced can be oxidized or used up in some other way. Remember, the NADH, which is the reduced form of NAD, carrying those electrons, should otherwise be sent off to the electron transport chain. But if the electron transport chain backs up, we have to find another route to oxidize this NADH so that we can recycle it and allow that glycolysis to continually occur. Okay, now as a sort of a price to pay or a byproduct of this process, we then synthesize lactate. And lactate is a small pathway. Let's actually go here. Oops. There we go. And so this small pathway is where we can shuttle pyruvate via the enzyme lactate dehydrogenase to create lactate. And so here we see that we can oxidize our carriers and recycle them back into glycolysis. So glycolysis will only occur as long as we can continually oxidize our carriers, which is use the electrons in some way, recycle those oxidized form of our carriers back to glycolysis where they can be reduced once again. And so as the lactate is produced here, this is where we actually get the burn in muscle. So if you think about, sprinting or running, if you're a sprinter or run really fast, or even heavy lifting, if you're a weight lifter in the gym, and you start to feel that burn in your muscles, it's the production of lactate, which is converted to lactic acid, that actually gives us this burn as it begins to build up. And what's actually happening on a deeper metabolic level is your rate of breathing is not matching the rate of ATP production via the electron transport chain. And so you're not bringing in enough oxygen to drive the electron transport chain. This is why it happens with sprinting, right? You're not breathing at a rate that's comfortable enough in order to drive oxidative phosphorylation, which needs oxygen. And so you can temporarily create ATP to sustain that type of workout, but you're going to have that price to pay, which is the buildup of lactate. And then that's going to be felt as that burn that you get in your muscles. Okay, just to give a little bit of clinical context to what this pathway is about. So let's move on to our second objective here. This is to discern between our body's different mechanisms of energy storage and use. So essentially, what are some of the other biomolecules that we can harness energy from? So we've talked a lot about glucose, but what are some other ways that we can use or store glucose in order to facilitate ATP production? So we've got carbohydrates, lipids, and proteins, and we'll talk about them individually. And so this is our main metabolic pathway. So these are several different pathways that are really just clustered into one giant cellular respiration pathway. And so we've talked about the main one in the middle here, glycolysis, the linking step here, which makes acetyl-CoA from pyruvate, the Krebs cycle or the TCA cycle, and then the electron transport chain where we have oxidative phosphorylation. So we've talked about the central part of this metabolic pathway here, but we can also oxidize or break down or catabolize other biomolecules as well. We can catabolize triglycerides, right? These are essentially glycerol backbones and those 3 fatty acid tails. We can break down the glycerol backbone via gluconeogenesis, and we can make glucose that can enter the glycolysis. And then we can break down those fatty acid chains or fatty acid tails by simply just grabbing the carbons off of those fatty acid tails and either shuttling them off into the Krebs cycle via acetyl-CoA or simply shuttling them off as other intermediates in between the Krebs cycle and and oxidative phosphorylation. In other words, our electron carriers, so NADH and FADH, can be produced that can go to oxidative phosphorylation. As far as our proteins, so proteins are broken down into amino acids. Those can be, broken down to liberate urea and ammonia. These have really important functions in our kidney. And then the other metabolites can enter cellular respiration at different points. So they can be converted into pyruvate, they can be converted into acetyl-CoA, or they can enter the Krebs cycle as other intermediates as a part of that cycle as well. And so this is how we can store energy in these other forms. So store energy in fat cells as triglycerides, store energy in proteins, right? This is pretty much muscle, where our proteins create the building blocks of muscle. Or we can simply store energy in the liver and in the kidney as glycogen, right? And this is where we go from glucose to glycogen. Remember, glycogen is going to be that more complex carbohydrate. So several glucose monomers stacked onto each other. And so we can break that larger complex molecule down to liberate glucose and drive glycolysis as well. Now, I want to make sort of a disclaimer here. Glycogen and triglyceride are our main energy storage and energy molecules that we can catabolize to make ATP or to drive this process, which then makes ATP. Proteins are not usually catabolized, at least not right away. You'd have to be in a starvation mode or in severe energy malnutrition. Think about kwashiorkor or marasmus, which are our protein energy deficient states. You'd have to be in one of those severe starvation states in order to start breaking down your muscle, okay, and liberating amino acids to drive ATP production. So just giving a small disclaimer here that while all three of these are plausible causes in our physiology, usually fats and glycogen are what are broken down to drive ATP production. Okay, so we've said quite a bit here. Let's kind of go into these a little bit more in detail individually. So starting off with glycogen metabolism, so we can store up glycogen in liver and kidney, and sometimes muscle as well. And we can essentially use that glycogen when the body's in a state of need and liberate glucose. Remember, glycogen is nothing but a bunch of glucose covalently bound to each other, as we can see at the bottom here. And so we can break this down, send that into the blood, just as if we were to eat a meal. We can use our own storage of glycogen to liberate that glucose into the blood and use it in our other cells to make ATP. Now, the caveat here is that only the liver and kidney have the enzyme that can allow this to leave the cell. So only glucose in its original state can leave the cell, go into the bloodstream, and be used by other cells. And so it cannot do so in this form, glucose 6-phosphate. It must be reconverted back to its original form, glucose, in order to leave the cell and be metabolized elsewhere. And so only the liver and the kidney have the enzyme that can do that. That is glucose 6-phosphatase. And if you remember me mentioning earlier, when we see ASC at the end of an enzyme, that means it's going to cleave a phosphate or cut a phosphate. And so in doing so, it removes that phosphate from glucose 6-phosphate. And we end up with our original form of glucose, which can leave the cell, right? Liver cells, kidney cells, go into the bloodstream and be circulated and utilized by our general system in our other cells, okay? We can also synthesize new glucose from some other substrates. This is called gluconeogenesis. So we can use glycerol coming from the breakdown of our triglycerides. We can use lactate, which is the buildup of that, process where we have glycolysis occurring absent oxygen. Or we can break down amino acids. Again, think about extreme starvation cases. But we can use these other substrates to create new glucose and to drive glycolysis as well, okay? This occurs mainly in the liver, and it's not just a reversal of glycolysis, but it's a separate series of enzymatic reactions with different enzymes altogether involved, okay? And you really don't have to focus on the details of memorizing this, but just understand that gluconeogenesis can use other substrates such as glycerol, lactate, and amino acid to generate new glucose that can be catabolized as a part of glycolysis, and then in turn drive cellular respiration as well. Okay, so this is illustrating that general idea. So we have glycogen up here. We've got these two processes, glycogenolysis and glycogenesis. So this is different from gluconeogenesis, just to make that disclaimer. So this process, gluconeogenesis, is different from this process here. So this is where we use glycogen, remember that complex carbohydrate, and we can break it down to glucose 6-phosphate. But in order for this to leave the cell, it must be converted to its original form, glucose, via the enzyme glucose 6-phosphatase. Okay, and so when it does this, it can then be, exported into the bloodstream and used in our other cells. And as I mentioned earlier, only liver and kidney cells have this enzyme that can do that. So only liver and kidney cells can store glycogen, reconvert it to glucose, and send that fresh glucose to our cells to be catabolized, okay? And this is what I mean by saying that it's not just a reversal of the initial process, but it's a separate pathway with different enzymes that are only present in some cells altogether. Okay, let's look at our second energy storage molecule here. This is our triglycerides. And triglycerides, we talked about the structure of them. Glycerol backbone with the 3 fatty acid, which are essentially these carbon chain tails attached. And so lipids actually have more energy per weight than carbohydrates or proteins. And I'm sure we can make this connection when we think about our diet and calculating the calorie worth of our food, right? When we look at per gram, carbohydrates and proteins give us 4 calories per gram, but fats give us 9 calories per gram. So fats are much more energy dense, and so they have more energy than our other 2 biomolecules. And so this is why they also play a more important role in terms of storing and utilizing energy in this way. So most of our triglycerides are stored in our fat cells. This is why people gain weight or get fat in certain areas, because you're simply creating more of these cells that are storing energy for later use. This is why it's so important for your energy input to balance or be matched to your energy output. Otherwise, you start to either dip into your protein stores, break down muscle on one end, or create excess adipocytes and adipose tissue on the other end. Okay, oops. Now, the process of breaking down fats to be metabolized is a little bit more complex, so let's kind of walk through this here. So we've got our triglycerides, and in the presence of an enzyme called lipase, again, ASC meaning that cleaving or cutting is happening, this can be broken down into those 2 components, that glycerol backbone with the 3 fatty acid, those carbon chains attached. And so the glycerol can do one thing, as we can see here. And then the fatty acids can do another thing, as we'll talk about in a minute. So the glycerol can actually be converted to DHP. And if you remember from glycolysis, when we talked about it initially, DHP is one of our glycolysis metabolites. So when I break down my triglyceride, I create glycerol that can be shuttled into glycolysis by the conversion of glycerol to DHP, dihydroacetone phosphate. Okay, and that can generate some ATP as well. In terms of what the fatty acid tails can do, they can be broken down by a process called beta oxidation. And here we essentially take those fatty acid tails, which are just carbon tails, right? Lots of carbon stacked or covalently bound to each other. And we can just grab those carbons off 2 at a time. And each of those carbons, each 2 of those carbons can be broken down into acetyl-CoA, NADH, and FADH. Acetyl-CoA can enter cellular respiration via the Krebs cycle, as we can see here. So this is acetyl-CoA coming from those fatty acids that can enter via the Krebs cycle. Our other carriers here, NADH and FADH, we know are the reduced form carrying electrons, and so they can enter cellular respiration via shuttling those electrons directly to the electron transport chain, as we can see down here, okay? And so each of those 2 carbons can actually generate 17 ATP for each of those 2 carbons. Okay, and so this is how we can take our triglycerides, sort of break it into its smaller components and start to metabolize them separately and use that to drive the overall process of cellular respiration, which the end goal is to make ATP as energy for our cells. Okay, the final molecule that we can oxidize or break down or catabolize here is proteins. And again, that huge disclaimer that I made earlier, this really only happens as a need basis in severe starvation states. So this process is called proteolysis, where we break proteins down into amino acids. And then from those building blocks, those amino acids, we can then convert that into several different substrates here. So we can convert that by a process called deamination. We remove the amino group, okay? And then we can send these substrates to either create pyruvate or enter the Krebs cycle via the production of acetyl-CoA. And so this is how we can allow those metabolites, those substrates, to then enter cellular respiration and drive ATP production as well, okay?